Welcome back to the Deep Dive, where we take the most complex biological blueprints and show you what actually matters.
Today, we're tackling something huge.
Yeah, the massive challenge of gene expression control. I mean, the mechanisms that decide which blueprints get built, where they get built, and when.
It's everything, really.
Yeah.
It's the difference between just having a library with all the instructions for every cell in your body, the genes for your liver, your brain, your skin, and actually being a liver cell.
A cell that has to ignore, what, 90% of its own instruction manual?
Exactly. It's the ultimate software that dictates the hardware.
And we're going to start with a classic Hollywood fantasy that really highlights why this control is so crucial. The Jurassic Park challenge.
Yes.
You remember the premise. Scientists get some T-Rex DNA from a mosquito stuck in amber, and poof, they have a dinosaur.
It's great cinema, but it illustrates this profound biological impossibility. And it's not just about the DNA being too old and degraded, which it would be. The real failure point is the control system. I mean, even if you could synthesize a perfect T-Rex gene, just dropping that DNA into a random spot in a modern reptile egg would be a complete failure.
Because you have the blueprints, but you don't have the construction crew or the schedule or any of the permits.
That's a perfect analogy. Gene expression is regulated by countless incredibly precise DNA elements, promoters, enhancers, all these boundary elements. If you just drop a gene into a new genome, it's going to be, well, completely out of context.
It won't know when to turn on or off.
Or it'll run at 10% capacity when it needs 100. This isn't just a problem for dinosaur movies. It's the core challenge in modern gene therapy. Getting a newly inserted, healthy gene to activate at the right time and at the right level. That's literally the difference between a cure and a failure.
So that's our mission today. We want to understand how cells, from simple bacteria all the way up to us, control this genetic machinery.
We're exploring the entire operating system of life, really. The system that ensures that while every single one of your cells holds the identical book of blueprints, that liver cell only expresses the genes for detox, while a muscle cell is busy with actin and myosin.
And we'll follow the complexity, right? Starting simple and building up.
Exactly. We'll start with the fast, efficient sort of single-layer control in prokaryotes, and then move into the multi-layered systems we see in eukaryotes. Transcriptional, processing, translational, and finally post-translational control.
Okay, so let's begin with bacteria. Their whole architecture is built for efficiency. It's a tight, ship-circular DNA, not a lot of junk, and really rapid response times.
And that genome organization is key to their speed. Genes that are involved in the same metabolic pathway are often clustered together, one after the other.
In a continuous sequence.
Right. And this clustering allows for what we call coordinated regulation. One single genetic switch can turn on or off all the enzymes needed for an entire process.
And their whole system is just a marvel of responding to the environment. They can't afford to waste energy, so they have to adjust enzyme production based on what food is available, like right now.
That environmental awareness is so beautifully shown in the classic experiments. Take lactose induction, for instance.
Okay.
If you have a bacterial culture that's just, ticking along, and you suddenly add lactose to its food source, within minutes, you see this incredible rapid spike in the production of beta-galactosidase mRNA.
The message to build the enzyme.
Exactly. And that's followed almost immediately by a huge surge in the protein itself. We're talking about a cell going from maybe 5 copies of the enzyme to about 5,000. A 1,000-fold increase in just a few minutes.
Wow. And that speed proves the regulation is happening on the fly, right, at the transcription level, not from some pre-existing stockpile.
Precisely. And you see the opposite with something they make themselves, like the amino acid tryptophan.
Right. This is tryptophan repression.
Yeah. If the bacteria are in a medium that doesn't have any tryptophan, they'll turn on the 5 genes they need to synthesize it. But the second you provide it in their food,
To shut the factory down.
instantly.
Yeah.
The system represses those genes because the cell knows it doesn't need to waste the energy anymore.
And this whole coordinated system is what Jacob and Monod first described back in 1961 as the operon model. So what are the basic parts of this little molecular machine?
The operon is a functional complex, and it has 4 main parts, all clustered together. First, you have the structural genes.
The blueprints for the actual enzyme.
Exactly. They lie right next to each other and get transcribed into one single long messenger RNA. We call it a polycistronic mRNA.
So one transcript carries the instructions for multiple separate proteins. Super efficient.
Very. Second part is the promoter. That's just the binding site where RNA polymerase docks to start transcription. Third, and this is crucial, is the operator.
The switch itself.
It's the switch. It's a DNA sequence that often overlaps with the promoter, and it acts like a parking spot for a repressor protein. If that repressor is parked there, it physically blocks RNA polymerase.
And the fourth part makes the repressor.
Right, the regulatory gene, which encodes that repressor protein.
So the whole game is controlling the state of that repressor, whether it's on or off.
Yes, and its state is controlled by the very substance the operon is designed to manage. Let's look at a repressible operon first, like the TREP operon.
Repressible meaning it's normally on. The cell is making tryptophan unless tryptophan itself turns it off.
Precisely. The TRP repressor protein is actually made in an inactive state. It can't bind to the operator by itself. So no tryptophan around, promoter is open, RNA polymerase goes to town, and the cell makes its amino acid.
But when tryptophan shows up...
When it becomes plentiful, it acts as a corepressor. It binds to that inactive repressor protein, changes its shape, and activates it. That new complex then just slams down onto the operator, blocking transcription.
A perfect feedback loop, high product, immediate shutdown.
Incredibly neat, and saves a ton of energy.
Okay, so what about the opposite system, the inducible operon, like the famous lac operon?
Right, this one regulates the enzymes for breaking down lactose. Here, the repressor is active by default. It's basically always sitting on the operator, keeping the system shut down.
So how does the presence of lactose turn it on?
Lactose itself, or really a derivative called allolactose, acts as the inducer. When it's present, it binds directly to that active repressor. This causes the repressor to change its shape, inactivating it and making it fall off the operator.
And with the repressor gone, RNA polymerase can get to work.
The operator's free, polymerase binds, and the genes are transcribed. The cell starts eating the lactose. Then, as the lactose gets used up, the inducer lets go of the repressor, which becomes active again and shuts the system back down. This whole thing is called negative control, because the regulator, the repressor, inhibits expression.
But the lac operon has that famous second layer of control. It won't touch lactose if glucose is available. This is where positive control comes in.
This is the glucose effect. It's brilliant. If you give a bacterium both glucose and lactose, it will burn through all the glucose first and completely ignore the lactose enzymes.
Glucose is the preferred fuel.
It is. So it represses the production of enzymes for other sugars, even if they're right there.
And this involves the signal molecule cAMP, which was a bit of a surprise.
It's a huge surprise. We thought cAMP was just a eukaryotic thing. But in bacteria, its concentration is inversely proportional to glucose. High glucose, low cAMP.
And low glucose means high cAMP.
Right. And that cAMP binds to a protein called CRP, the cAMP receptor protein. This complex is the key to positive control.
So why does this complex need to bind to the lac operon?
Because the promoter for the lac operon is actually, well, it's pretty weak. RNA polymerase doesn't bind to it very efficiently on its own.
It needs a boost.
It needs a big boost. The cAMP-CRP complex binds to a DNA site just upstream of the promoter. When it binds, it physically bends the DNA in a way that dramatically helps RNA polymerase to dock and start transcription.
So you need two things to be true.
Two things. Lactose has to be present to get the repressor off. A&D glucose has to be scarce, so you have high cAMP to get the activator on. It's a perfect biological A&D gate.
That makes perfect sense. Now, going back to tryptophan for a second, there's another, even more subtle layer of control called attenuation.
Attenuation is.
Yeah.
It's really elegant. It's a way to stop transcription after it has already started.
A secondary brake pedal.
A secondary brake, yeah. When tryptophan levels are high, the brand new mRNA transcript folds into a very specific shape, a hairpin loop, that acts as a termination signal. It literally knocks the RNA polymerase off the DNA before it even gets to the structural genes.
Wait, how does the RNA know what the tryptophan level is?
This is the genius part. The very beginning of the mRNA, the leader sequence, has a couple of codons for tryptophan in it. When tryptophan is scarce, the ribosome that's translating this leader sequence stalls because it's waiting for the right tRNA.
Okay.
That stalling physically prevents the RNA from folding into the terminator hairpin. Instead, it folds into a different, non-terminating shape, and the polymerase keeps going.
But if tryptophan is abundant,
the ribosome zips right through the leader sequence without stalling, which forces the RNA to fold into that terminator hairpin.
Yeah.
The actual speed of the ribosome dictates the RNA shape, which then controls the gene. It's incredible.
it's like primitive wiring. And that leads us to riboswitches, which take the protein out of the equation entirely.
Right. Riboswitches are parts of the mRNA itself, usually in the 5-foot untranslated region, that can bind directly to small molecules like adenine or vitamin with super high specificity.
So the metabolite literally changes the shape of its own mRNA.
Yes, The binding causes this massive conformational change in the RNA, and that new shape directly blocks gene expression. It might form a hairpin that terminates transcription, or it might hide the sequence the ribosome needs to start translation.
So it's an all-RNA control system.
No protein cofactors needed. It's RNA acting as its own independent regulatory machine. A really profound idea, and maybe a little window back into the ancient RNA world.
It's just so clear that nature invented the first programmable microprocessors with these systems. This ability to use inputs to get binary on or off outputs is, well, it's the foundation of synthetic biology.
Absolutely. The whole goal of synthetic biology is to use cells as these microscopic, programmable robots. Engineering immune cells to hunt down specific cancers or getting bacteria to make biofuels. But to do that, you need a logic circuit.
And as we just said, the lac operon is a ready-made ANE gate. Expression happens when lactose is present and D-glucose is absent.
Exactly. So scientists can take these natural parts promoters, repressors, operators, and start mixing and matching them to build their own custom genetic networks. The problem is it's insanely complex.
You have to get everything perfectly tuned.
Perfectly. The affinity of the repressor for the operator, the expression level of the repressor itself, it's like trying to build a computer chip by hand, one transistor at a time.
Which is why we now have computer-aided design, or CAD systems, for this.
Right, this was pioneered by Christopher Voigt's group. The idea was to take synthetic biology from a sort of tinkering hobby and turn it into a real engineering discipline.
So the engineer doesn't need to worry about the nitty-gritty DNA sequences. They just design the logic.
That's the goal. They can specify the logic they want, maybe a circuit with a few A and D gates and an OR gate using standard symbols. The CAD software then goes into a database of pre-characterized parts and automatically picks the DNA sequences that will produce that behavior. It's a fundamental shift in how we approach engineering life.
Okay, so now we have to make the big pivot. We're leaving the streamlined world of prokaryotes and moving into the much, much more complex world of eukaryotes. And here, regulation means dealing with a huge physical barrier, the nucleus.
That compartmentalization is, in itself, a huge regulatory step. In bacteria, transcription and translation happen at the same time, on the same molecule.
Right.
In eukaryotes, separating them with the nuclear envelope allows for all these extra layers of processing and quality control before the message ever gets to a ribosome.
So let's break down that barrier, the nuclear envelope. It's way more than just a simple membrane.
Oh yeah, it's actually two parallel membranes, separated by a tiny gap. And the outer membrane is physically continuous with the rough ER, and it's even studded with ribosomes.
And is connected to the outside.
It is. It has these specialized proteins, nesprins, that literally link the outer nuclear membrane to the cytoskeleton, which helps anchor the whole nucleus in place.
And what about support from the inside?
That's the job of the nuclear lamina. It's this thin, dense meshwork of proteins called lamins, which are a type of intermediate filament. The lamina sits right under the internuclear membrane and gives it mechanical support, helps it keep its shape, and also acts as an anchor for all the chromatin inside.
And we know how important this is because of what happens when it breaks. The clinical relevance here is just stark.
It is. Mutations in the gene for lamin A, the element AI gene, cause a whole spectrum of diseases. The most famous and severe is Hutchinson-Gilford progeria syndrome, or HGPS.
So rapid aging disease.
Yes, characterized by incredibly rapid premature aging. And if you look at cells from these patients under a microscope, their nuclei are just a mess. They're misshapen, lobulated, fragile. It's a clear demonstration of the lamina's structural role.
And the mutation itself is such a subtle, devastating example of regulation gone wrong.
It's often caused by what seems like a harmless synonymous mutation. It doesn't even change the amino acid. But what it does is create a new, incorrect splice site in the RNA. This leads to a shortened, toxic version of the protein that just wrecks the entire nuclear architecture.
A tiny error in RNA processing with catastrophic results. So with this envelope as a barrier, how does anything get in or out? This brings us to the nuclear pore complex, the NPC.
The NPC is less like a pore and more like the world's busiest customs checkpoint. The amount of traffic is just staggering. An actively growing cell has to import something like 500,000 ribosomal proteins every single minute.
Every minute. That's insane. What does this checkpoint even look like?
It's a monster. One of the largest protein complexes in the cell. And it has this beautiful octagonal symmetry. It's built from about 30 different proteins called nucleoporins.
And what makes it so selective? How does it stop just anything from getting through?
The central channel is lined with a special type of nucleoporin that has these repeating sequences of phenylalanine and glycine, so we call them FG repeats.
Okay.
These repeats form this disordered, flexible, hydrophobic meshwork. You can picture it like a dense tangle of oily spaghetti. This mesh physically blocks the free diffusion of anything big, anything over about 40 kilodaltons.
So big molecules need a passport to get through that oily mess.
They do. For a protein to be imported, it needs to have a nuclear localization signal, or NLS. The classic one is a short stretch of positively charged amino acids. This NLS tag is recognized by a transport receptor in the cytoplasm.
And that receptor is the cargo truck.
It is. It's a protein dimer called important alpha beta. The cargo, with its NLS, binds to the important. The whole complex then docks onto the MPC, and it moves through that central channel, sort of hopping between those oily FG domains.
So once it gets inside the nucleus, what makes it let go of the cargo? And what stops the important from just going right back out?
This is where the molecular engine comes in. It's a small GTP-binding protein called Ran. The cell maintains a massive concentration gradient of Ran. There's tons of Ran-bound to GTP inside the nucleus and very little in the cytoplasm.
And how's that gradient maintained?
By two accessory proteins, one called RCC1 is stuck in the nucleus, and it acts like a recharger, converting Ran-GTP to Ran-GTP. And another, Ran-GT1, is in the cytoplasm, and it does the opposite. This steep gradient is the energy source that gives the transport its direction.
So when the impult complex hits that high-energy nuclear environment,
The high concentration of Ran-GTP in the nucleus binds to the important, and that binding causes the whole complex to fall apart, releasing the cargo. The important, now bound Ran-GTP, gets exported back out, where the GTP is hydrolyzed and the cycle starts again.
and for export, it's the reverse.
The reverse. Ran-GTP actually promotes the assembly of export complexes. It's a beautiful, simple system for ensuring traffic only goes one way.
All right, stepping inside the nucleus, we hit the next big problem, the one the envelope was designed to contain, the packaging problem. You gave us the numbers. 6.4 billion base pairs in 46 chromosomes have to fit inside a 10-micrometer nucleus.
It's like trying to coil 10 miles of thread into a tennis ball. It requires these incredible multiple levels of compression. And it all starts with the fundamental unit, the nucleosome.
Which is built from histones, these small, basic, and incredibly conserved proteins.
Astonishingly conserved. Histones H3 and H4 are almost identical between peas and cows. And that's because they have to interact with the DNA backbone, which is, universally identical and negatively charged. And the first level of packaging is just wrapping the DNA around an octamer of these histones.
And this was figured out with a really clever digestion experiment.
Yeah, Kornberg's lab showed that if you digest chromatin with enzymes that cut DNA, you don't get random fragments. You get these repeating fragments of about 200 base pairs. This implied that something was protecting the DNA at regular intervals.
The nucleosome core.
Exactly. The core structure is 146 base pairs of DNA wrapped almost twice around a disc-shaped histone octamer, which is 2 copies each of H2A, HB, H3, and H4.
So where does the fifth histone, H1, fit in?
H1 is the linker histone. It sits on the outside of the core, kind of like a clamp, binding where the DNA enters and exits the nucleosome. It helps lock everything down. If you remove H1, you get that classic beads on a string looking electron micrographs.
And this first level of coiling gets you a packing ratio of about 7:1.
Right. And when we finally got the crystal structure of the nucleosome, we saw those crucial flexible tails sticking out from the core.
They weren't just random bits.
No, not at all. While the main body of the histones forms this globular structure, these N-terminal tails project outward. And we now know they're the primary targets for all the enzymes that regulate chromatin. They're absolutely critical.
You also mentioned histone variants, so there are different flavors of these proteins.
Yeah, specialized versions for specific jobs. For example, CENPA replaces the normal H3 only at the centromeres. Another one, H28X, gets phosphorylated at sites of VNA breaks and acts like a molecular flare to recruit the repair machinery. It shows that packaging is not just about stuffing DNA in, it's functional.
Okay, so 7:1 is a start, but we need something like 10,000-to-1 for a mitotic chromosome. So how do we get to the next level, the 30-nanometer fiber?
The string of nucleosomes has to fold up on itself into a thicker fiber. The most accepted model is that the nucleosomes stack up in a kind of double helix. This step gets the total packing ratio up to about 40-to-1.
And those tails you mentioned are essential for this step.
Absolutely essential. For example, the tail of H4 from one nucleosome has to reach out and interact with the H2A, H2B dimer of a neighboring nucleosome to pull them together. If you chop off those tails, The chromatin can't form that 30-nanometer fiber.
And then even that gets further organized.
Right. That fiber is then gathered into these large, supercoiled looped domains, which are tethered to a protein scaffold. And the mitotic chromosome is just the ultimate, most condensed state of all of this.
And this physical state, condensed or open, is what divides chromatin into two functional classes, euchromatin and heterochromatin.
Euchromatin is the dispersed, loose, transcriptionally active stuff. Heterochromatin is the highly condensed, compacted, and transcriptionally silent state.
And there are two types of the silent state.
Right. Constitutive heterochromatin is permanently silenced. It's mostly repetitive DNA near telomeres and centromeres. Then there's facultative heterochromatin, which is DNA that has been specifically inactivated in a given cell type.
And the classic example of that is X chromosome inactivation.
The bar body, exactly. In female mammals, one of the two X chromosomes has to be completely shut down to equalize the dosage of X-linked genes with males, who only have one X.
And the choice of which one to shut down is random?
It's random in the early embryo, but once that choice is made, it's permanent. It's passed down to all the daughter cells. And this is why you get genetic mosaicism, like the beautiful patchwork coat of a calico cat. Those patches are clones of cells that inactivated one X or the other.
So how does a cell silence an entire chromosome?
It starts with a long non-coding RNA called XIST. The X bytes RNA is transcribed from the X that's going to be inactivated, and it literally coats that entire chromosome, spreading out from its source.
And that coding kicks off the silencing process.
It initiates it. But the long-term stable silencing is maintained by establishing these repressive epigenetic marks, mainly extensive DNA methylation and specific histone modifications.
Which brings us to one of the most important concepts in all of this, the histone code. The idea that patterns of chemical modifications on those histone tails actually encode information.
That's the core hypothesis. These modifications, methylation, acetylation, phosphorylation, act in two main ways. First, they can be docking sites for other proteins that come in and read the code. Second, they can physically change how the nucleosomes interact with each other.
Like acetylation.
Right. Acetylating a lysine on a histone tail neutralizes its positive charge. This weakens its grip on the negatively charged DNA and also prevents it from interacting with an adjacent nucleosome, which physically opens up the chromatin and makes it more active.
So let's walk through establishing a silent state. How do you set up a repressive mark, like methylation on histone 3, lysine 9H3K9?
Okay, so if a region is active, that H3K9 is probably acetylated. The first step to silencing it is to bring in an enzyme called a histone deacetylase, or HDAC, to remove that acetyl group.
Wiping the slate clean.
Exactly. Once that's gone, a histone methyltransferase comes in and adds a methyl group to that same lysine 9.
And that methyl group is the new signal. It's the off switch.
It's the off switch. And that specific mark, H3K9 methylation, is a binding site for a protein called HP1, heterochromatin protein 1. HP1 binds to the methyl mark, and then HP1 recruits more of the methyltransferase.
So it creates a positive feedback loop.
A self-propagating wave of silencing that spreads out from the initial site, compacting the chromatin as it goes.
We also see small RNAs playing a role here, guiding this machinery.
Yeah, especially in silencing repetitive DNA. It seems that small RNAs that match those repetitive sequences can guide the whole repressive complex to those specific locations to make sure they stay shut down. It's like a genomic surveillance system.
Okay, speaking of the whole chromosome, we need to touch on the structure of the mitotic chromosome and the large-scale aberrations you can see with karyotyping.
A karyotype is just the standard picture of a cell's chromosomes, arrested in mitosis, organized into homologous pairs, and ordered by size. With modern staining, you can see these beautiful banding patterns, and large-scale screw-ups are immediately obvious.
And these can be caused by things like X-rays. Let's talk about translocations.
A translocation is when a piece of one chromosome breaks off and attaches to another non-homologous chromosome. The most famous clinical example is the Philadelphia chromosome.
In chronic myelogenous leukemia, CML.
Right. It's a shortened chromosome 22 that results from a swap with chromosome 9. This swap fuses the ABL gene from chromosome 9, which is a potent kinase that drives cell proliferation, with the BCR gene on chromosome 22.
And the resulting fusion protein is the problem.
The BCR-ABL fusion protein is basically the ABL kinase stuck in the on position, driving uncontrolled cell division. It's the direct cause of the cancer.
The text also points out a translocation was key in human evolution.
Yes. The reason we have 23 pairs of chromosomes and other great apes have 24 is because two ancestral 8 chromosomes fused end-to-end to form what is now our chromosome 2, a massive chromosomal change that helped drive speciation.
Amazing. Okay, let's go to the ends of the chromosomes, the telomeres.
Telomeres are the protective caps. They're just thousands of repeats of a short DNA sequence T tag G in humans, and they're essential. They stop the ends from being degraded and, crucially, stop the cell's repair machinery from thinking the end of a chromosome is a broken piece of DNA and trying to fuse it to something else.
And they solve the end replication problem.
The fundamental problem.
Yeah.
Because of how DNA polymerase works, with RNA primers, every time a linear chromosome is copied, the very end of the new strand is a little bit shorter.
So without a fix, your chromosomes would shrink with every cell division.
They would. And the telomere also has this 3-foot overhang that folds back on itself to form a protective loop, which is stabilized by a protein complex called shelterin.
And the fix for the shrinking is the enzyme telomerase.
Discovered by Blackburn and Greider, telomerase is a special reverse transcriptase. It makes DNA from an RNA template, and it carries its own little RNA template inside it.
So it's a self-contained extension machine.
It is. It binds to that 3' overhang and uses its RNA template to add new teaching-edge repeats to the end of the chromosome, extending it. Once it's long enough, normal DNA polymerase can come in and fill in the other strand.
And this has huge implications for aging and cancer.
Massive. In most of our adult cells, telomerase is turned off. So our telomeres do shorten as we age. This is the Hayflick limit. Eventually they get so short it triggers cell death or senescence, which is a good thing. It's a break against cancer.
But cancer finds a way around it.
About 90% of all human cancers have figured out how to turn telomerase back on. That's what gives them the immortality they need to keep dividing forever.
It's a longevity clock. Finally, let's look at the other specialized region, the centromere.
The centromere is that constricted region where the kinetochore assembles during mitosis to pull the sister chromatids apart. It's defined by the presence of that special histone variant we mentioned, cENPA.
And the really surprising thing here is that the centromere is not defined by its DNA sequence.
This is a major shift in thinking. The DNA sequences at centromeres are not conserved at all, even between closely related species. This tells us that the identity of the centromere, its location and function, is an epigenetic trait.
It's defined by the presence of the cENPA chromatin structure, not the A's, T's, C's, and G's underneath it.
Exactly. It's another powerful example of how cell identity is maintained by information layered on top of the genetic code.
So for a long time, we pictured the nucleus as this, bag of spaghetti, just a random soup of chromatin. But now we know it's incredibly organized.
Highly structured. We know that chromosomes don't just float around randomly. They occupy distinct, non-overlapping regions called chromosome territories.
Like little neighborhoods.
Exactly like neighborhoods, and it's not random. For instance, gene-poor chromosomes, like chromosome 18, tend to be pushed out to the edge of the nucleus near the lamina, while gene-rich chromosomes, like 19, are more in the center.
But it's also dynamic. Genes have to move and interact, sometimes even across different chromosomes.
And this is where the new technologies like chromosome conformation capture or Hi-C have just blown the doors off. They let us map which bits of DNA are physically close to each other inside the 3D space of the nucleus.
And they've revealed these amazing interchromosomal interactions.
They have. For example, if you treat breast cancer cells with estrogen, two of its target genes, which are on completely different chromosomes, one on chromosome 2 and one on 21, will rapidly move from deep inside their territories and come together in close physical proximity.
Which supports the idea of transcription factories.
It does. The idea that active genes are physically moved to these preassembled hubs of transcription machinery for more efficient processing. The data from Hi-C has also revealed another layer of organization called TADs, or topologically associated domains. These are regions of the genome that preferentially interact with themselves, like self-contained folding units.
And within this organized space, we also have these specialized membraneless compartments.
Right, these nuclear bodies that form through phase separation. The best studied are called speckles. They look like irregular blotches, and are basically storage depots for splicing factors.
So when a gene turns on and needs to be spliced,
the factors are mobilized from the nearby speckle and travel to that active gene. You can literally see them move. It's a dynamic supply closet.
Okay, before we dive into the last few layers of control, we need to come back to that foundational idea we started with. Every differentiated cell has the complete, identical genome.
Right, and this was proven by those classic cloning experiments. Stewart showing a single carrot cell could grow into a whole plant. Gurdon taking the nucleus from a tadpole gut cell and growing a whole new frog.
And of course, Dolly the sheep, which really cemented the idea for everyone.
Dolly was cloned from a mammary gland cell of an adult sheep. And these experiments proved two huge things. One, differentiated cells don't throw away genes. And two, that differentiated state is totally reprogrammable. The cytoplasm of an egg has factors that can wipe the slate clean and start development over.
So cell identity isn't about which genes you have, but how you use them. And that use is governed by four main levels of control.
A whole hierarchy. You have transcriptional control, which genes get turned on, then processing control, how that RNA is spliced, then translational control, if and when that mRNA gets made into protein, and finally post-translational control, how long that final protein sticks around.
Let's start at the top. Transcriptional control. This is the main on-off switch in eukaryotes. And to study it, we first needed tools to see which of the 20,000 genes are on in any given cell.
And the first big breakthrough there was the DNA microarray, or DNA chip. This lets you look at thousands of genes all at once.
You spot DNA for each gene onto a slide, and then you use color coding to compare two different cell types.
Exactly. You take mRNA from, say, a cancer cell, and you convert it into cDNA and label it green. You do the same for a healthy cell, but label it red. Then you mix them together and wash them over the chip.
A green spot means the gene is on in the cancer cell. Red means it's on in the healthy cell.
And yellow means it's on in both. The brightness tells you how much. It was revolutionary for seeing these big, coordinated shifts in gene expression.
But microarrays have now mostly been replaced by something even more powerful: RNA sequencing, or RNA-seq.
RNA-seq is a huge leap. Instead of hybridizing to pre-selected spots, you just take all the RNA in a cell, convert it to cDNA, and sequence everything directly. It gives you a much deeper, more accurate picture of everything that's being transcribed.
Including things microarrays would miss.
Like non-coding RNAs, new splice variants. It's incredibly sensitive. It's used all the time now in the clinic, like for profiling breast cancer tumors, to decide on the best therapy.
Okay, so the actual molecules that do the controlling here are the transcription factors, or TFs.
Right. These are the proteins that bind to the DNA regulatory sites. And the key in eukaryotes is that the control is combinatorial. A single gene needs a whole specific committee of TFs to bind to it to be expressed correctly. And conversely, a single TF might help control hundreds or thousands of different genes.
This combinatorial logic is what creates all the different cell types.
It is. And it's why some TFs are called master regulators. If you take a fibroblast, a generic connective tissue cell, and you force it to express the TF called MyoD, it will turn into a muscle cell.
It rewires the whole program.
It does. Or expressing the eyeless TF in a fly's leg can cause an eye to grow there. It's incredible.
And the most profound example of this is making induced pluripotent stem cells, or iPS cells.
Yamanaka's work, showing that you could take an adult skin cell and by adding just 4 specific transcription factors, you could reprogram it all the way back to a pluripotent embryonic-like state. It proved that the epigenetic state that defines a cell is completely reversible.
So let's look at the architecture of these TFs. How do they actually read the DNA sequence?
Most of them bind as dimers, and they have these specific structural motifs that fit into the major groove of the DNA double helix.
The most common one being the zinc finger.
A zinc finger is a little protein domain that's stabilized by a zinc ion. And these TFs often have a whole series of these fingers, which allows them to wrap around the DNA and recognize a longer, more specific sequence.
Then there's the basic helix-loop-helix, or BHLH motif.
These proteins have to dimerize to function. The helix-loop-helix part is what lets them pair up, often with a different BHLH protein, which creates a huge amount of diversity. The basic region right next to it is what actually touches the DNA. MyoD is a BHLH protein.
And finally, the leucine zipper, or BZIP.
Here, the dimerization is driven by a coiled coil structure, where you have leucines at regular intervals that let 2 alpha helices zip together. And again, an adjacent basic region makes the contact with the DNA.
Okay, let's look at the DNA sites they recognize. The PCK gene is a great example of how complex this is.
The PTCK integrates all these different metabolic signals. So, right at the start, you have the core promoter with the TATA box, which positions RNA polymerase. Just upstream, you have proximal promoter elements like the CAT box that bind more general TFs and control how often transcription starts.
And then you have the really specific stuff further away, the distal response elements.
Right. These are the binding sites for hormone receptors, like the glucocorticoid response element or the insulin response element. This is how the gene listens to all the different signals in the body and integrates them into a single transcriptional output.
And with all these sites scattered across the genome, how do researchers even find them?
The main technique is called chromatin immunoprecipitation, or ChIP-seq, usually followed by sequencing, so ChIP-seq.
Okay, walk me through it.
First, you use chemicals to cross-link the TFs to the DNA they're bound to, right inside the cell. Then you break up the chromatin into small pieces. Next, you use an antibody that's specific for your TF of interest to pull down only those DNA fragments that are attached to it.
And then you sequence that DNA.
And that tells you every single place in the entire genome where that TF was bound. It's an incredibly powerful way to map these regulatory networks.
So let's look at an activation mechanism in detail. How does something like the glucocorticoid receptor work?
Okay, so the GR sits inactive in the cytoplasm. When the hormone cortisol comes into the cell, it binds to the GR. This binding causes a shape change that uncovers the receptor's NLS.
The nuclear import tag.
Right. So the whole complex moves into the nucleus, finds a glucocorticoid response element in the DNA, and binds as a dimer, activating transcription.
And it can do this even if that binding site is really far away from the gene's promoter through an enhancer.
Enhancers are amazing. They can be thousands of base pairs away, upstream or downstream, even flipped upside down, and they still work. They do this by causing the DNA to loop around, bringing the activator proteins bound to the enhancer into direct physical contact with the machinery at the promoter.
And that physical link is often made by these huge complexes called coactivators.
Right. And coactivators do two main things. Some of them act as a bridge, physically connecting the activators to RNA polymerase. The mediator complex is a key example of that.
And the others actually modify the chromatin.
Exactly. Because you can't transcribe a gene if it's wrapped up tightly in a nucleosome. So the first job is to open up the chromatin. The activators recruit coactivators that are histone acetyltransferases, or HATs.
They add acetyl groups to the histone tails,
which neutralizes their positive charge, loosens their grip on the DNA, and generally decondenses the chromatin, making it accessible.
Once it's open, the next crew comes in, the chromatin remodeling complexes.
Right. These are big machines that use ATP to physically shove nucleosomes around. They can slide a nucleosome along the DNA to uncover a promoter, or even kick the histone octamer right off the DNA entirely, creating a nucleosome-free region where the transcription machinery can assemble.
And we see a very specific pattern of this around active genes.
We do. Active promoters almost always sit in one of these nucleosome-free regions, or NFRs, flanked by two very well-positioned nucleosomes. And we also see specific histone modification signatures. Acetylation is high right at the promoter, but a different mark, H3K36 methylation, is found along the body of the transcribed gene, and we think that helps prevent transcription from starting in the wrong place.
So we've covered how to start transcription, but sometimes the cell needs to be ready to go instantly.
That's the idea behind paused polymerases. For a lot of genes that need to be activated really fast, RNA polymerase is already bound at the promoter. It's even started making a tiny bit of RNA, but it's stalled by inhibitory factors.
So activation is just releasing the brake.
It's just removing the inhibitor, often by phosphorylating it. This allows for a much more rapid response than having to recruit and assemble the entire complex from scratch.
Okay, what about the other side of the coin? Transcriptional repression.
It's often the mirror image of activation. So instead of HATs, you have HDACs, histone deacetylases, that come in and remove those activating acetyl groups. This allows the chromatin to condense back down.
And the ultimate way to lock a gene down for good is DNA methylation.
This is the covalent addition of a methyl group directly onto cytosine bases in the DNA, specifically at CpG sequences. This is a very stable, heritable epigenetic mark that is strongly associated with long-term silencing.
It's how the inactive X chromosome stays inactive.
It is, and it's also responsible for a weird phenomenon called genomic imprinting.
Where a gene is expressed or not, depending on which parent you inherited it from.
Exactly. For example, the IGF-2 gene is only expressed from the copy you get from your father. The copy from your mother is silenced by methylation. This is fragile, and if something goes wrong, it can lead to diseases like Prader-Willi syndrome.
And finally, let's circle back to the long non-coding RNAs, but this time as repressors.
This is a really cool mechanism. The LNCRNA HOTAIR is a great example. It acts as a molecular scaffold.
A bridge.
A bridge. One end of the HOTAIR RNA binds to one repressive protein complex, and the other end binds to a different repressive complex. It then guides this whole double repressor machine to a target gene on a completely different chromosome and shuts it down. It's transacting regulation guided by RNA.
Okay, let's pivot to the plant world for a minute for just a beautiful, simple model of how this combinatorial control works to build a structure. Let's talk about flower development.
Flowers are built from 4 concentric rings, or whorls. From the outside in, you have sepals, petals, stamens, and carpels.
And this was all figured out by looking at mutants where, say, a flower grew petals where its stamens should be.
Right. These homeotic mutants suggested there was a simple genetic code dictating the identity of each whorl, and that led to the ABC model.
Proposed by Meyerowitz. It uses just 3 classes of genes A, B, and C in combination.
And the logic is super simple. The A gene alone gives you sepals on the outside. Where A and B overlap, you get petals. Where B and C overlap, you get stamens. And C alone in the center gives you carpels.
So if you knock out the B gene,
you lose petals and stamens. The A function expands inward, and the C function expands outward. So you get a flower that goes sepal, sepal, carpel, carpel. It's a perfect demonstration of combinatorial TF action.
All right, once transcription is done, we're still not finished. The RNA has to be processed, and this is the second major level of control, mainly through alternative splicing.
Alternative splicing is a huge sort of complexity. It's how one gene can make multiple different protein products. It's a major reason why our proteome is so much more complex than our genome.
A simple example is fibronectin.
Right. The version of fibronectin made by liver cells and secreted into the blood is different from the version made by fibroblasts for the extracellular matrix. And the only difference is that 2 specific exons are included in the fibroblast version, but spliced out in the liver version.
And the most extreme example is the DSCAM gene in fruit flies.
It's mind-boggling. The DSCAM gene helps wire the fly's nervous system. It has these 4 different clusters of exons, and in the final mRNA, you have to pick just 1 exon from each cluster, which means you can make a staggering number of different proteins.
The math works out to over 38,000 different possible protein isoforms from that one single gene. It's how you can get the specificity needed to wire a complex brain.
So how does the cell choose which exons to include?
It's controlled by regulatory proteins that bind to specific sequences in the RNA called splicing enhancers or splicing silencers. If an activating protein binds to an enhancer, it helps the splicing machinery recognize and include the exon. If a repressive protein binds a silencer, the exon gets skipped.
And beyond splicing, there's also RNA editing.
This is where the cell goes in and chemically changes a base in the RNA after it's been made. A key human example is the apolipoprotein B mRNA. In the liver, the full-length protein is made. But in the intestine, an enzyme comes in and changes a single C to a U.
And that change creates a stop codon.
It creates a premature stop codon, resulting in a much shorter protein that has a completely different function related to fat absorption. A single letter change with a huge physiological consequence. Okay, the mRNA has survived the nucleus and splicing. Now it's in the cytoplasm, ready to be translated. This is our 3rd level of control.
And here, the key regulatory sites are the untranslated regions, the UTRs, at the 5 and 3' ends of the message. A lot of mRNAs are held in this stored inactive state, ready to go at a moment's notice.
Like right after fertilization.
Exactly. An unfertilized egg is packed with maternal mRNAs that are kept translationally silent, often by a protein called maskin that loops the mRNA into a circle binding to both the 5' cap and the 3' end, which prevents the ribosome from getting on.
And fertilization breaks that loop.
Fertilization triggers a signal that releases maskin, the poly A tail gets lengthened, and you get this massive burst of translation to kickstart development.
And this control can also happen globally, like during stress.
Right. If a cell is stressed by heat shock or a virus, kinases will phosphorylate a key initiation factor called EIF2. This effectively shuts down almost all protein synthesis to conserve energy.
Then there are very specific controls, like with ferritin and iron levels.
Ferritin is the protein that stores iron safely. When iron levels in the cell are low, a protein called the iron regulatory protein, IRP, binds to a sequence in the 5' UTR of the ferritin mRNA.
And physically blocks the ribosome.
It's a physical roadblock. The ribosome can't start translation. When iron levels are high, the iron binds to the IRP, making it fall off the mRNA, and now translation can proceed. So you only make the storage protein when there's actually iron to store.
We should also just quickly mention cytoplasmic localization.
Right. Cells can physically move mRNAs to specific locations before they're translated. This is critical in development. In the fruit fly egg, the bicoid mRNA is shipped to the anterior pole, and the Oscar mRNA is shipped to the posterior pole.
Using zip codes in their 3' UTRs.
Exactly. This ensures that when the proteins are made, they are already in the right place to set up the head-to-tail axis of the embryo.
The last piece of this is mRNA stability. How long does the message even last?
This varies wildly. The mRNA for a regulatory protein might only last a few minutes, while the mRNA for hemoglobin can last for days. And this lifespan is mostly determined by the length of the poly A tail.
And degradation starts with shortening that tail.
Right. Enzymes called deadenalyses are always nibbling at the tail. Once it gets critically short, the message is targeted for destruction, either by being decapped and degraded from the 5-foot end in structures called P-bodies, or by being chewed up from the 3-foot end by a complex called the exosome.
And this whole process is heavily regulated by microRNAs.
miRNAs are these tiny, non-coding RNAs that bind to the 3-foot UTR of target mRNAs. And their most common job is to speed up their destruction. They recruit the deadenylase enzymes, accelerating the decay of the message. They are master fine-tuners of gene expression.
And that brings us to the end of the line, the final level of control, which deals with the protein product itself. Because sometimes you need a protein to do its job and then disappear fast.
Absolutely.
Yeah.
This regulated destruction is carried out by a machine called the proteasome. It's this big, hollow, cylindrical complex. The inside of the barrel is lined with proteasomes.
So how does the cell mark a specific protein for destruction? How does it tell the proteasome what to eat?
The kiss of death tag is a small protein called ubiquitin. A protein that's targeted for destruction gets a whole chain of these ubiquitin molecules attached to it.
And that polyubiquitin chain is the signal.
It's the signal. A family of enzymes called ubiquitin ligases are responsible for recognizing specific target proteins and adding this chain. They're incredibly specific. They'll tag a misfolded protein or a cell cycle protein whose time is up.
And once it's tagged, it's off to the proteasome.
It binds to the cap of the proteasome. The ubiquitin chain is clipped off and recycled. The protein is unfolded using ATP, and then it's fed into the central chamber and chopped up into small peptides. It's the cell's garbage disposal and regulatory timer all in one.
So we started today with the sheer impossibility of the Jurassic Park challenge. Just getting the right genes to turn on in the right cells at the right time.
And we've seen how life solves that problem by putting checkpoints at almost every single step of the process.
From the physical packaging of the DNA as heterochromatin through the complex logic of transcription factors and the histone code.
All the way to alternative splicing, miRNA-mediated decay, and finally deciding the exact lifespan of every single protein with the proteasome. The depth of this combinatorial control is it's astonishing.
And this brings us back to that final thought on cloning and cell identity. The experiments prove that the genetic information, the DNA sequence, is identical and is reversible.
So the information that really defines a cell, what makes the liver cell different from a neuron, isn't the DNA sequence itself. It's the epigenetic blueprint. It's the pattern of histone modifications, the DNA methylation, the stable structural cues like the centromere's location, this inherited non-sequence-based information. That is the critical layer that scientists are still racing to fully understand.
And that holds the real key to the ultimate control of life.
I think it does.
A perfect thought to end our deep dive on gene expression control. Thank you so much for walking us through all of this complexity.
It was my pleasure.
And thank you for listening. Until next time.